This is Chapter 6, Segment 1, pages 185 to 193. The first section is titled Everything You Know is Wrong, and this talks about long delay learning. In previous rigorous experiments with rats and runaways had indicated that learning was extremely difficult if the intervals between the response and reward delay was longer than just a few seconds. In the eye blink conditioning, it shows optimal learning when the trace interval between the CS and the US is less than a second. This suggested a qualitative difference between flavor aversion learning and other types of associative learning. The surprising aspect of taste aversion learning is that only certain combinations of CS and US could be learned. For example, when a flavor was paired with an illness, it worked well, but when the flavor was paired with an electric shock or an audiovisual cue was paired with an illness, no evidence of learning occurred. This combination of the two was crucial to influence learning. Mechanisms evolved to deal with specific problems that animals face in the wild emerged. Taste aversion learning is the evolution's solution to the rat's problem of learning to avoid foods that contain slow-acting poisons. Taste aversion learning also occurs in humans. This questions if the principles generalize from one principle to the other in humans. The next section was special characteristics of flavor aversion learning. And in one-trial learning, a potent flavor aversion can be learned in one-trial conditioning. This shows the results of an experiment used in rats, which a novel saccharin drink was associated with an immediate injection of lithium chloride. The two groups tested averaged nearly zero consumption of the saccharin after only one saccharin-lithium pairing. Consumption increased quickly when the saccharin was offered again without the lithium. And this displays extinction. One of the groups was given a delay of 28 days between the third extinction trial and a test. And there was a slight spontaneous recovery of the aversion. Conditioning is fast, and extinction and spontaneous recovery are not that different from typical forms of classical conditioning. On long delay learning, the delay between the flavor and the US that allows flavor aversion conditioning. Sam Revsky first noticed that the methods used in taste aversion learning experiments were special. He had an explanation for why they might allow learning over such long delays. A rat that learns an aversion to saccharin over a 1-hour delay in the typical lab experiment has little to do with the delay interval. The rat does not eat anything for that 1-hour interval. It usually sleeps, and it wakes up knowing that it was nothing else to be blamed but the saccharin. Revsky captured this idea by emphasizing stimulus relevance and interference, and this is important in conditioning. Stimulus relevance, when for a given US only some kinds of CSs are good or relevant predictors. When illness in the US, the most relevant CS is a flavor, and the rat therefore learns about the flavors when it gets sick. Revsky's interference idea is that relevant stimuli tend to compete with one another for conditioning. If extra CS, or like second flavors, are introduced during the delay interval, the 1-hour period between eating and feeling sick, for example, there's less conditioning to the saccharin or to the poison. This is because the rat is unsure which CS to attribute the poison to. In the learned safety section, the flavor aversion learning is explained as an evolved so that the omnivores could discriminate foods containing poison from foods that are safe to ingest. This was protective evolution, and rats might also judge lights and noises the same way. Callatt and Rosen suggested that this had a role in long delay learning. Revsky's theory implies that an aversion could form over an infinite delay if there were no interfering relevant flavors, but such an aversion does not typically form. In delay interval, the rat is gradually learning that the flavor tasted is safe. Over time, this gets in the way of learning an aversion if illness does not eventually happen. Michael Best believed that in aversion learning, learned safety would actually be analogous to conditioned inhibition. If the learned aversion is extinct, then safety is inhibition. Best first showed that conditioning inhibition can be produced in flavor aversion learning. In the hedonic shift section, hedonic shift is described as the main effect of aversion learning is to change the palatability of the flavor from pleasant to noxious. Several investigators have used this interesting method called the taste reactivity test, based on the fact that rats react differently to flavors that differ in palatability. When exposed to tasty sucrose solution, they show a set of yum behaviors, and this could be protruding their tongues and licking their paws. When exposed to bitter flavors, they display a yuck response, and this is they rub their chin on the floor and walls, they gape, and they shake their paws.